Hey everyone, and welcome. So you've got that big photosynthesis test coming up, right? Well, you are in the perfect spot. We're going to treat this like a workout session, designed to get you in shape to absolutely crush those end-of-topic exercises. Let's do this.
Look, it is totally normal to get a little overwhelmed by these exam questions. You've got graphs to interpret, variables to figure out, all these definitions to remember. It can feel like a lot. But seriously, don't sweat it. We're going to go through every single part, piece by piece, so you can walk into that exam feeling totally confident.
All right, here's our game plan. We're going to start with some quick-fire questions to get warmed up. Then we'll make sure we've mastered all the key terms. After that, we'll take a deep dive into some data, decode a few experiments, and finish up by absolutely nailing the diagrams. Sound good? Let's go.
Okay, first up, the quick-fire round. This is all about multiple choice and comparison questions. These are designed to test how well you know your core facts and how fast you can recall them. So let's get those brains warmed up and ready to go.
Let's zip through these. For 1.1.1, the key is remembering respiration happens all the time, but photosynthesis, that needs light. Simple as that. For 1.1.2, you just have to know that a photosynthesizing plant is going to be pumping out oxygen. Now, 1.13 is all about ATP. Think of it as the rechargeable battery of the cell, carrying energy from the light phase to the dark phase. For 1.1.4, this is a classic trick question. Oxygen is a product of photosynthesis, not a factor that changes its rate. And finally, for 1.1.5, you just have to carefully read the deductions, and you'll see that the first three are spot on.
All right, for these AB questions, you have to be super precise. The energy-storing molecule, that's ATP, so it's A only. The organelle for photosynthesis, you know this one, it's the chloroplast, so B only. Where's the chlorophyll stored? In those stacks called grana, so A only again. Now, watch out for this one. Where does the light-dependent phase happen? It happens in the grana, but that's not an option here, is it? So the correct answer is none. See how they try to catch you out? And finally, the gas plants release is oxygen, O2, making it A only. Getting these details right is how you score big.
Let's move on to Section 2, Mastering Key Terminology. Honestly, this is your superpower. If you know these definitions cold, you can unlock what any exam question is really asking. First up, photolysis. Here's a little trick to remember it forever. Just break the word down. Photo means light, and lysis means to split. So photolysis literally just means splitting with light. What's it splitting? Water. Easy, right? Next, the stroma. It's so important to connect a structure with what it does. The stroma isn't just some goo inside the chloroplast. It is the specific location for the light-independent phase, or the Calvin cycle. This is where all the action happens to actually build the glucose. Okay, rapid-fire round. Here are the other must-know terms from this question. I'd say pause right here and try to define each one out loud. We've got chlorophyll, photosynthesis, blue-black, ATP, starch, chloroplast, iodine solution, and enzymes. Knowing these is absolutely non-negotiable if you're aiming for a top grade.
All right, let's kick it up a notch. Section 3 is our data deep dive. This is where we stop just memorizing stuff and start thinking like actual scientists. We're going to interpret the results of a real experiment on growing lettuce.
Rule number 1. Before you even glance at a graph or a table, you have to know the aim. What was the point of the whole experiment? Here, it's super clear. They wanted to see how changing temperature, light, and CO2 would affect how fast the lettuce grew, which they measured by its mass. The lettuce's mass is basically a stand-in for the rate of photosynthesis. Okay, so the data is like a story, and we have to read it. What's the deal with light intensity? What things are holding back the process, the limiting factors? And how did the scientists get the absolute best results? Let's dig in. So this is a perfect example of what we call limiting factors. At first, not enough light is the problem, but once you give the plant plenty of light, something else becomes the bottleneck. In this case, CO2 and temperature. It's like baking a cake. You can have all the flour in the world, but if you run out of eggs, you can't make any more cake. To get the fastest growth, you have to find that sweet spot where everything is just right. This is exactly what commercial growers do in their greenhouses to get maximum yield. Now, this is a top-tier, high-flyer kind of answer. It's not good enough to just say it gets too hot. To get the best marks, you have to explain why. Photosynthesis is run by enzymes, which are like tiny biological machines. If you heat them up too much, they denature. They lose their shape and just stop working. That's the science behind it. Okay, on to Section 4,
time to put on our imaginary lab coats. We're going to dissect how experiments are designed, looking at everything from pondweed bubbling in water to identifying variables and making sure our results are reliable. So check out this data from the pondweed experiment. They're measuring how long it takes for the plant to release 20 oxygen bubbles. Now think about it. A shorter time means the plant is working faster. So a quick look at these numbers tells you that blue light is working really well, and green light, well, not so much. And when you turn that table into a bar chart like this, the result just pops. It's so obvious, right? The lowest bar is blue light, meaning the fastest rate. The tallest bar is green, the slowest. This is why we visualize data. It makes the conclusion impossible to miss. Blue light is the winner here. Okay, this is so important for any science class. The independent variable is the one thing you decide to change on purpose. In this experiment, that's the color of the light. The dependent variable is what you measure to see the effect of that change. Here, it's the time taken to release those 20 bubbles. Change one thing, measure the other. That's the core of a fair test. These little details are what examiners love to see. Why do you wait 5 minutes before starting to count? You've got to give the plant a chance to get used to the new light conditions. How can you make your results more reliable? Repeat the whole thing a few times and find an average. And here's the big connection. Why do leaves look green to us? Because that's the color they don't use much. They reflect it away right into our eyes. This experiment proves that theory perfectly. All right, we're in the home stretch. Section 5, our final section, is all about diagrams. And here's the good news. You do not need to be an amazing artist to get full marks. You just have to be clear, accurate, and follow the rules.
Seriously, just think of it as a checklist for easy marks. 1, give it a caption. Bam, 1 mark. 2, draw the right shape. A basic oval with a double membrane around it. Don't forget that second line. Another mark. 3, label at least 3 parts you know well. Stroma, granum, lamella. Perfect. Label them clearly with straight lines. You do that, and those 5 marks are all yours.
This is a classic procedure you've just got to memorize. It's a 4-step recipe. Step 1, boil in water. This breaks down the cell walls. Step 2, boil in alcohol. This gets rid of the green chlorophyll so you can see the color change. But, and this is a huge safety point, alcohol is flammable, so you must use a water bath, never put it directly on a flame. Step 3, rinse in cold water to soften the now brittle leaf. Step 4, add your iodine and look for that color change. And just like with the chloroplast drawing, getting the marks here is all about following the rubric. Give it a good caption, draw the outline of the leaf. Now, the key part. You only shade in the area that was exposed to light, that cross shape, because that's the only place photosynthesis could happen and make starch. Then just label the shaded bit blue-black and the unshaded bit reddish-brown. It's that straightforward.
Wow, we have covered a lot of ground today. Everything from multiple choice tactics to data analysis. But remember, the real goal here isn't just to memorize these specific answers. It's to build your skills so you can tackle any question they throw at you. If this helped you out, do me a favor and share it, like it, and subscribe for more. And for even more practice, be sure to follow Mr. Try on all his social channels. Now go get ready for that test. You are absolutely gonna nail it.